Okay, and just you can wait me 1 second. Yes, in the previous class, we were discussing about cancer stem cells, and I also shared a video which I wanted you to follow for the last class. And I'm going to quickly go through it anyway. So, in case you missed it, you should be able to get through it. So, the point is that the two models are, one of it is a stochastic model, and the other one is cancer stem cell model. In stochastic model, the assumption is that it is to explain the heterogeneity, one of the ways, heterogeneity, and also about the ability to form tumors. Is it that any of the cell in a tumor can cause tumor or not? So in the stochastic model, each of them may have any of the cells you can take or cell type, and it will keep on growing. And different cell types are given rise to by different cells, cell type, progenitors like things, or each of them has the potential to give rise to a tumor. Whereas in cancer stem cell model, the idea is that the whole cancer is kind of a tissue, which we will also discuss about tissue organization today, which will be useful in understanding the general architecture too. In the whole tissue, like tissue, the tumor has some stem cells which keep on giving rise to other cell types, right? That is the essence of cancer stem cells. In that sense, only a few cell types, few cells can actually give rise to a tumor, while the others cannot give rise to tumor. That is the cancer stem cell model. It is relatively well accepted too. So once again, what happens is that in the cancer stem cell model, we are learning about, sorry, yes, here we are, and the we also read about it, and liquid tumors, and we have the solid tumors. Liquid tumors is about liquid tissue, animal, sorry, the blood and lymph and those kinds of things come under liquid tumors, leukemia and so on. And the solid tumors are majority of the other tissues. So what we know of it is, at least in one sense, we will discuss in more detail, that say, for example, 1 in 1,000 cells, if we took 1,000 cells and out of them about one of it is a cancer stem cell, that means the other 999 are just tumor cells. These do not or cannot have the capacity to give rise to a tumor. They cannot. Whereas this cancer stem cell can give rise to a tumor. So in different tissues, we have different proportion of the cancer stem cells. We will see a table eventually. And the idea of why we are studying the cancer stem cell is, or what happens during therapy is, we have it, if there is a tumor, we try to go with chemotherapy, and we try to go with radiotherapy or so. At least chemotherapy and radiotherapy, many of them do target the genome, and genome is broken down, because of which the cells, if they divide, will produce non-productive cells, and then they will die out. Whereas when this person is treated with them, after some days the tumor comes back. There is a relapse of the tumor, and why is that happening? And then therefore we are not able to treat the tumors properly, or the cancer properly. So if this is a tumor here, and then we target we treat with chemotherapy and radiotherapy, and then we kill majority of the cells, the rapidly dividing cells, but not the cancer stem cells, because cancer stem cells are actually slow growing compared to the rest of the tissue cell types. And then when we treat it with chemotherapy or radiotherapy, the rapidly growing cells, these, all of them, they would die. Whereas the cancer stem cell, because it is slow growing, that means when it says slow growing, it means that if the damage is inflicted, then there is enough time for repair to occur. And then it will get normal. So cancer stem cell is kind of protected from it. And then after a while, after the treatment, the cancer stem cells give rise to a tumor. So the idea is somehow to be able to target the cancer stem cells so that the cancer stem cells can be eliminated, and the other cells would be eliminated anyway through senescence, aging, and death eventually. So did we discuss this part? I would just want you to tell me about it. Adult intestinal, I mean, did we discuss about this?
No,
Somebody?
sir.
No, right?
No, we haven't.
Okay. So if you take any of the adult stem cells, any tissues, I was talking that we will discuss about the organization. Every tissue has stem cells. Those are adult stem cells here. And those adult stem cells, when there is a need, I think we discussed about the tissue number homeostasis like this, right? The adult stem cells will keep producing these functional cells, and the functional cells will die after wearing and tearing and so on. So the number will be constantly maintained as such by occasional reproduction by the adult stem cells. They will give rise to and try to fill in the dearth of the cells. So adult stem cells may undergo asymmetric cell division, because of which one of it is self-renewal, and the other one is a transit amplifying cell, or you can call it as TAC. The full form is transit amplifying cell. That means we call it transit because it is between stem cells and the completely functional, between the completely functional cells or differentiated cells. So this transit amplifying cells, amplifying, it will divide rapidly and then form eventually progenitors and finally completely differentiated cells. These are the cells that actually perform the functions, in this case, intestinal. So their intestinal lining would require different types of cells, and those are produced by the intestine, the stem cells within the epithelium, and they will divide into rapid transit amplifying cells, which will eventually give rise to these. So if you can try to see the organization, something like this, general organization of majority of the tissue systems is this, and one, you can think of it just like one asymmetric cell division will give rise to one another cell type, isn't it? Like the transit amplifying cells. These transit amplifying cells will rapidly divide. They will keep on dividing further like this. And eventually they will give rise to progenitor cells, you can think of. And each of those progenitor cells will give rise to functional cells. We call these, this is amplification is happening here, right? In this case, we have the first differentiation. I mean, not first differentiation as a result of this asymmetric cell division by the adult stem cell. And that one cell is dividing multiple times, but eventually, or within 3 rounds of replication or so, as in this case, each of them are giving rise to functional cells, right? One cell division by the stem cell can result in multiple number of cells. That is why there is amplification happening here. That's why we call these cells as transit amplifying cells. So this is
So,
yes, please.
can TACs renew themselves or they have to mature as differentiate from
Which one? Which
SCs?
one?
TACs.
Yes, transit amplifying cells cannot undergo beyond a certain number of cell divisions. Here I've shown 3, right? Probably 2 is maximum sometimes. And as the amplification is happening, this is a stem cell here, right? And the stem cell, and here we have the differentiate, or here there should have been the differentiated cells. Say I would just put like this. Okay, so that means the property of being stem cell, that is the stemness, is reducing, and as it is dividing more and more, it becomes more and more differentiated. So cell is changing from one type to the functional terminally differentiated cells. So between this region, the transit amplifying cells will undergo cell division, but they are usually not. They're not. I don't think they are asymmetric cell divisions, and they could be a little bit, but eventually they will divide twice or 2 times or 3 times, 3 rounds maybe, and then they will give rise to, ultimately they will not remain in transit amplifying state for too long. Okay, did I answer your question, Rishi?
Yes, sir. So basically, they
Yeah.
undergo very little of self renewal and differentiate easily.
Yes, you can say that. Yes.
Okay.
Okay. Or it is something like this. We cannot probably use the word self-renewal as such, because once it forms, rapidly it divides and eventually forms the terminated cells, terminal differentiated cells. There is probably no way of culturing these as transit amplifying cells, or they won't remain as transit amplifying cells. One asymmetric division, they will form, and that's it. That is how the design of the tissue is. But what you said, in one sense, can it makes some meaning too. Yes. Okay. So all the tissues, I don't need you to mug up all the names of these things. I want you to understand the organization of the tissues. And the same is the case here with hematopoietic stem cells. Here we have the hematopoietic stem cell, which can divide into either forming these CLP or CMP, and then some of them will give rise to the red blood cells, macrophages, and so on. The others will give rise to those predominantly involved in immunity, and so on. So if you go through different names, different tissue types, they may give several names to based on the tissue type, which I'm not actually bothered about at this time. We don't need to mug up those things right now. So the same thing can happen with tissues, with cancers also. You can take, this is a hematopoietic stem cell, which will divide into myeloid progenitor or lymphoid progenitor. Based on that, this will give rise to lymphoid tissue types, and here these are the red blood cells or blood cells. And here you can see that there is amplification kind of thing, and also differentiation to give rise to different cell types. That is why we call hematopoietic stem cell as a multipotent stem cell, because it's giving rise to many different cell types. In leukemia, which is a cancer of the blood, what can happen is, because of a mutation, it will give rise to another type of leukemic stem cell, this is called leukemic stem cell, LSC. And then it gives rise to some tissues which will, some cell types which will divide and eventually produce leukemic blast cells. So if we can probably kill the leukemic stem cells, probably we won't have these for too long. They will die out eventually. One of the issues that you need to also consider is the end product of the normal cells is functional cells, right? But this may not be true in majority of the cancers. What I mean is, in when there are mutations, they may not be terminally differentiating into these kinds of cells. They will just form non-functional cells. If you see it in that sense, what does this cell? So we will talk about blood. and I will just give about 1,000 cells. We want them all functional. So there is a hematopoietic stem cell that keeps replenishing, and as and when they are worn out, they will die. That's the general organization of the cell number homeostasis. And in if there is a mutation and leukemic leukemic stem cell has formed, it will the blood, say for example 100 cells have died. Okay, there are 900, and some of the cells have become leukemic stem cells. They will keep producing cells, but they are non-functional. What does the body feel? It's like the body would want more cells, so some kind of signal can be reaching to the stem cells, and they will keep on producing more and more. And along the same signals can also be improving or allowing the production of leukemic stem cells, but they're producing non-functional cells. So that is another point you have to remember about a cancer. How is a cancer different from that of a normal tissue? Two things we can remember is unregulated cell growth is one point, or cell division. And the second one is non-functional. The produced cells are non-functional. They don't perform any function. Their function is probably just to divide more and more. So that causes twice a problem. First thing, they're abnormal growth, invading into other spaces, tissues, resources. And the second thing is all those cells are non-functional, contributing nothing to the body. So with this, I would also want to discuss about origins of the cancer stem cells. I'll switch to a different color. So as I was discussing about, we have the stem cells, and the stem cells will give rise to progenitor cells. You can also put transit amplifying cells in there. And then we will eventually have, these are transit amplifying cells, assume that, and then they will give rise to a differentiated cell. These are functional. So where do these cancer stem cells originate? So this requires a bit of a discussion that I will try to discuss. Say stem cells, and the stem cells will give rise to transit amplifying cells, and the transit amplifying cells will proliferate rapidly, and they would eventually give rise to differentiated cells, right? What does, if we remember, what I was discussing about is, in mutations lesson, we have discussed that all mutations happen spontaneously, or I mean, many mutations happen spontaneously. If there is replication, there is a good probability of a mutation, right? So in which case we would want to count on the rate of replication, not just replication. Why rate is, if there is enough time, then repair can occur, right? If there is no time between replication to replication, then the repair mechanisms cannot or may not have enough time to repair the DNA. So the mutations get established very easily. So if you see, there are three different cell types, right? Cell type 1, stem cell type 2, the transit amplifying cells, and cell type 3, the differentiated cells. In which case do you think, this is a general question and I hope you will participate with me, in which situation do you think will the one second, in which of these cell types do you think there is more probability of mutation, and why? Anybody?
Is
Anyone?
it the third?
Yes. In here? Why do you
Yes,
think so?
because it's there's a lot of changes happening. You're expressing one factor more than the other. You're silencing a particular gene and expressing one higher. So That's where
Okay.
methylation and other modifications happen. So
Yes.
that's
And
probably yes.
so what you said is certainly a possibility also. I want to think about where is the probability of mutation high? In which case do you think the mutation probability is high? Mutation can occur anywhere, okay? If not through replication, it can also happen because of induced mutation. So any of these any of the mutation will occur in all the cases. That is a possibility. What I'm asking here is, where do you think the probability is very high spontaneously? Probability of spontaneous mutations. It has to do with the rate of replication. In which time, which place do you think the rate of replication is very high?
TAC second.
This is the case. That's right. In stem cells, they're usually quiescent or they're dormant. When there is a need, they would divide and then give rise to transit amplifying cells, and they would divide much more rapidly. The differentiated cells usually do not have that much of, not much of replication to happen there. But a mutation, that means the stemness, if you see, there is very poor stemness in differentiated cells, there is a little bit, and there is full here. But these cells are very dormant. Embryonic stem cells are dormant. Stem cells are dormant. These are rapidly dividing one, in which case we have the highest rate of replication. That means there is a possibility of increased rate of cell mutations also. So at this time, what we can think, where do cancer stem cells come from, is it can be any of them. A mutation in the stem cell can cause a stem cell to become a cancer stem cell, or there is a mutation that could influence the transit amplifying cells mutation, and then it can give rise to a cancer stem cell. And a differentiated cell, terminally one, may get a mutation in which it is stopped, in whose the cell cycle was halted from regaining the cell cycle capacity and then undergoing cell divisions. So there are all possibilities, but in my opinion, I think this is the biggest probability is in transit amplifying cells. But anything is possible though. Okay, there are three properties similar to that of cancer stem cells, embryonic stem cells, or any of the stem cells. First one is self-renewal. Then second is, I will compare it with normal stem cells, okay? Stem cells also have self-renewal. The cancer stem cells also have self-renewal capacity. Stem cells, we know we differentiation, and here tumor differentiation within a tumor, that is by heterogeneity, that is more than one cell type. How is that happening? It is because of differentiation capacity of cancer stem cells. There is another one that is the ability to give rise to tumors. So we can derive these properties by looking at their ability to induce tumors, and we can do the self-renewal capacity by testing is by serial transplantation, spear formation. In vitro would be spear formation. Establishment of tumor heterogeneity can be dealt by, or can be proved by, flow cytometry. The point of this slide is, why we are learning about properties is, somebody, if they give you a sample of tumor and they ask you, it has three different cell types A, B, C, and the question is, if you want to prove if any of these cell types have cancer stem cells, are cancer stem cells, one should then be able to establish the tumorigenicity, the self-renewal capacity, and the ability for tumor heterogeneity. So the first one can be, first thing we will take is, we will take these bits of or some parts of the tumor, and we will put it in the mice, and we will use KID mice, that is severely combined immunodeficient mice, because if you take a human sample and put it in mouse, it might be immunograft rejection would occur. So we would have to choose a mouse in which has compromised immunity. So we put it subcutaneously under the skin, and then we will see if it can give rise to a tumor or not. If it gives tumor, then we know that one of these cell types has the ability to give rise to tumor, but we don't know which one it is. Then what we would do is we would have taken these cells, I mean the sample, we would have made a single cell suspension, and then we would have done FACS to sort them. That means we would get the 3 different cell types into A, B, C, different tubes. Now we can test. Here what we did is we took a small part of the sample and put it directly. And that means we put all the 3 A, B, and C cell types. Here now we can perform a correct experiment, is that we can give C cells to one type of mouse, B to another, and A to another. All of them always SCID or immunocompromised lab animals. And now we check which one has the ability to give rise to a tumor. Maybe A did not give, maybe B gave, and C did not give. That means out of these 3 cell types, A does not have tumorigenicity capacity, C does not have tumorigenicity capacity, and B has tumorigenicity. So that is how we prove one of the characteristics, the ability to give rise to tumors. That is the first thing. And we can also do, say, for example, we have done this experiment here, right? We have given, and a tumor has formed, and now we take this tumor and do FACS again. If we did FACS and we found that there are 3 different cell types, like in A, B, C again, it is proving something. We gave only cell type B, but we got a tumor, and within the tumor we again have 3 different cell types, A, B, C. That is proving the heterogeneity capacity. Okay, and assume that if you have put in, when we took the sample B, in this case sample B, and we have given the cells, say we gave 100 cells into the test animal, and if we got 1,000 cells, how is it possible? I'm writing here. We gave 100 cells, and upon transplanting it into the test animal, we got 1,000 cells. It would be possible if only there is a capacity for self-renewal. Right? You have cell division, and cell division should give rise to one cell type and one probably differentiated cell. Right? So if you had 100 cells and it went on cell divisions, at the end probably you will have 100 cells and 100 stem cells and 100 differentiated cells in one round of these things. They can also go symmetric cell division, where 100 of these cells can give rise to same cell type. So you will have 200 cells. Both ways you are proving self-renewal capacity. You can think of it, if the cell did not have self-renewal capacity, it would have given rise to, say, A cell type and, say, for example, C cell type. In this case, how many B cell types are left? There is 0, because the B cell type, if it divided into 2 differentiated cells, there are 0 B cells, right? That would be a proved lack of self-renewal capacity. You can keep on doing this. Say we took sample, we put it in mice, we got a tumor. Again, we take B cells from the tumor, and again, you transplant into different tissues like this. For several rounds, you can do it. that is meant by serial transplantation. Serial transplantation is an in vitro way of proving that the test cells have the self-renewal capacity. In vivo test is spear formation. That means it's, if you remember something like the embryoid body, and the embryoid body is when we take embryonic stem cells and we do perform by hanging drop method, they will form embryoid bodies like this. And the similar one can be the similar one can also be done using 1 second, I'll try to use this space. Yes, same thing can also be done in vitro, like you provide the culture conditions and you give the cancer stem cells or say, and they can form into small spears, and those are called as the spears in general. Okay, and they can form a spear, and then you can do FACS, see what all cell types are there. In that, you can take the cell type B, as in the previous slide I have shown, and again see if they can form the spears. And their ability to form a spears is a proof for self-renewal capacity. Okay, this is the same thing what is presented here. A tumor sample is given, and that is made into single cell suspension, and then FACS, cell sorting, and then we can read based on the cytogram or the graph that we get. Here we are seeing two different cell types, A and B. Okay, so first thing what we know is that the tumor that we got is heterogeneous. So it has, say, for example, A and B cell types. We don't know which of them is a cancer stem cell and which of them is not. So we should have taken A cells and give to a skid mouse, and B cells to another skid mouse, and see if they can form a tumor or not. If they can form a tumor, then it is the B cells are the cancer stem cells, and A or not. Okay. So if we keep on doing, and again, I didn't mention here what is CD4 expression and CD44 and CD24 here. So we will come across these things again, and so I will discuss at that point how to the markers for cancer stem cells. Here is a graph that the table that I want you to understand well, and I only have, what you need to know is tumor type, where is it isolated from, and injection is where exactly did we put in the mouse. So different cancers can proliferate well in different sites of the body. So brain ones, if they are in brain, they can go well. And then these are in the test animals, okay, where the transplantation is done to study the properties of these cancer stem cells. And this is about number of cells tested. That means how many cells are transplanted or injected, and whether a tumor is formed or not. And here is a report on the lowest number required for a tumor to form. So that means you take, there is a tumor, this is a tumor, assume, and it has several cells, and then you make a single cell suspension of it, single cell suspension, and then you do an assay like this. You give 100 cells to a mouse and see whether tumor is forming or not. You give 1,000 cells, 10,000 cells, or say 6 or 8 cells. And so, and then they will see whether tumor has formed or not. Here you might have formed, it might have shown given a tumor, this one also, and this one did not form. When you give only about 100 cells, probably the tumor has not formed. What does that say? It says that about at least one cell in these 1,000 cells is there to give rise to a tumor. right? And here 10,000, at least one cell is there in 10,000, at least one in 10 to the power 8 cells is there to give rise to a tumor. So, but we count this one, which will say the concentration of the cancer stem cells. That means at least 1 in 1,000 cells is a cancer stem cell. That's how we can say. Okay, that's why this is important. So the others may have higher number, but the proportion would be proportion wise. The 10,000, this sample may have about 10 cancer stem cells, and this one may have something like somewhere around that. Okay, 10 to the power. How much would that be? You're saying 1 in 100, so 10 to the power 6. Is that right? Maybe. So we have to first, when we look at this table, I would also want you to look at the type of the cancer that is there here, brain, prostate, and so on. In some of them, they have given at different locations, like subcutaneous below the skin and in prostate. And where is it going well is the question here. Same way, hepatocellular one, and it's given in intrahepatic and subcutaneous. Say we'll just concentrate on prostate cancer here. So they have number of cells is they have given 100,000, 5,000, and 10,000 probably. And what they have seen here is about least number of cells required to give rise to or for the tumor to form is about 100. At this point, I don't remember what is A mean, but to be able to interpret the data, we should be able to understand what is given here. We'll just focus on that. Prostate cancer cells are given to the prostate, and cells tested were from 100 to about 10 lakhs. And the least number required is about 1,000 cells. So in which of these tissues do you think the tumor is forming easily? Anybody? In which case do you think the tumor is forming easily? Looking at these numbers, you can say, even though you give 100 cells, the tumor is forming in subcutaneous. Here, you should give, in the prostate, you should give at least 1,000 cells for a tumor to form. So the tumor that we are looking at here is forming more easily in subcutaneous one. That's not the point of what I want to discuss anyway. But what says it is at least 1 in 100 cells is a cancer stem cell. That's what the meaning of this part is. Okay. So that is how you look at this data, or that is what the people do when they are experimenting. So in vitro assay is sphere formation. We discussed that already. Okay, I have I'll take 10 minutes, 5 minutes, and I would want to discuss a few things as a precursor for the next class. But otherwise, I would want you to watch the video that has been posted yesterday as well. So how to identify cancer stem cells? So if we have sorted out, first of all, in the previous one, we also used the CD44. I think it was given. I didn't discuss, but we will discuss later on. Sorting. If we get a sample of a biopsy sample of a tumor, and then we need to separate them, and different forms of can be used, like FACS. You want cell sorting to be to happen, but based on what properties should we do that? In the earliest times, we didn't know what properties we should search them. Just the forward scatter and side scatter are not many times not sufficient. There must be some properties that are different, specific to the cancer stem cells as opposed to the other tumor cells. Some property that should be there. It can be cell surface marker, or it can be some other property. So people wanted to find out those things, and in the earliest, one of the ones is using a dye called Hoist dye. and it is through efflux. So the central idea of this is we have several cell types in the cells. Say these are three different cell types, A, B, and C, and we treat them all with a dye, and based on their absorption or the uptake of the dye, retention within them, they will have, some might be high uptake, some will be medium, and the other one could be no uptake of the dye, right? And then we perform FACS, and then we can separate these cells into three different tubes. And then we can test which of these is the cancer stem cell, which of these has the cancer stem cell property. Now with advanced, as we progress, we started the experiments with something using the Hoist dye, and now people we know is we have several cell surface markers with that are being used. We will discuss that later on. So the Hoist dye is simple, that we add the dye, and the dye will permeabilize, or it will move into the cell, and some of the cells may have efflux pumps. So the dye that has moved in is kicked outwards like this. Okay, this is the dye. The dye molecule is kicked out. So if this efflux did not happen, this cell would have been fluorescent because the dye is inside. Whereas in some of the cells, the dye is entering into the cells, but it is effluxed out, so there would not be no fluorescence. Right? Based on those properties, we can separate the cells based on the fluorescence using FACS. It can be separated, and then we would probably get the A and B cell types into two different tubes, which can then be tested for whether they are fluorescent, whether they are cancer stem cells or not. When they performed such an experiment, what they found is some three different cell types. Here is one whole cell type, and these are some other cells, and there are some other cells, the ones that are shown in red here like this. So this is called as side population, because they're forming in the side or something. In this graph, they are on the side as dots. Each dot is a single cell count, a droplet passing through, and the reading that is obtained from the drop in FACS, using FACS. So these are called side populations, and people have given these cells and checked if they can form the tumors. No tumor. They have given these cells to these cells as well. No tumor. And they gave the side population cells into mice. Then they found a tumor formation, which means that there must be the cells in the side population are likely the cancer stem cells. Okay. When we are studying about cancer stem cells, I would want you to focus on trying to know how to figure out which one is cancer stem cell and which one is not. It is a technical, and which is involving about your research aptitude, right? How would you do it? So the design of experiments, that is what you should practice. Or if a data was given, then you should be able to figure out which one is the cancer stem cells. Okay, I will just introduce a few things here. This is called as ABCG. It is a ATP binding cassette efflux pump. This is the efflux pump that is called as ABCG. ATP binding cassette G is one, probably two efflux pump. Some of the cells, especially those that are in the side population ones, they have higher expression of ABCG, and because of which it is effluxing the dye. so it doesn't get stained as much. And so we don't know yet if ABCG is anyway related to the stemness in the cancer stem cells. Maybe it is not related at all. All we can say at this stage is ABCG is the one that is causing less amount of labeling by this dye, Hoy's dye. Okay, we will discuss. There is an interesting discussion about it in the next class. We will discuss in the next one. Okay. Yes. Any questions? Did you watch the video that I sent yesterday? I'll stop the recording now.